Students, in the last video lecture we have discussed how the blood enters into the liver. To understand how circulation in the liver occurs inside the liver, we have to understand the anatomy of the liver. A little bit of the anatomy we'll discuss. And this was the liver which we have discussed. If you have not gone through the first lecture, please go through my first video which is connected to the liver. This was the liver, and we know that hepatic artery from the aorta, a split of artery occurs which is called hepatic artery, enters into the liver and it takes oxygenated blood. On the other hand, we said there is portal vein which enters through spleen, through GIT and through the pancreas into the liver. It is having lesser amount of oxygen, or you can say it is a deoxygenated blood which is taken by the portal vein inside the liver. Now, anatomically, liver consists of lobes. We can differentiate three lobes. Inside the lobes, there are lobular structures are there, or lobule structures are present. These lobules are polygonal structures. These are polygonal structures. Correct? Polygonal structures which are present in the lobes of the liver. Right? We will understand this lobular structure in the lobes of the liver in a larger sense. Let us make a larger diagram to understand what this lobule structure is like. So I said it is a polygonal structure. I am making hexagonal structures. To make you understand. So these are the corners of the lobules. In center of the lobule, remember, liver consists of three lobes. Each lobe contains lobule structures. These are also called as hepatic lobules. Hepatic lobules, in a larger sense to understand, these are polygonal structures. In the center of this polygonal structure, what is present? A central vein is present. A central vein is present. On the each of the corner, what is happening in each of the corner? What is happening? A sinusoid. Sinusoid is a tubular hollow space which is entering from the each corner towards the central vein. Towards central vein. Remember, these are sinusoids. The sinusoids are hollow tubular structures which are entering from each of the corner in towards the central lobule. Towards the center. So, sinusoid means this is an empty vascular space or empty hollow space in a tubular structure which is called as sinusoid, entering from each of the corner towards the central vein. Now, in the sinusoid, the blood, which blood? You remember, this is the portal vein and this is the hepatic artery. Hepatic artery. So blood from these which is entering into the liver actually has come over here, come into the sinusoid. Let's use the colored pen to understand it. So one portal vein which is opening inside the sinusoid of the lobule of the liver, and hepatic artery which is taking the oxygenated blood which is also entering into the sinusoid. So these two bloods are entering inside the sinusoid of lobule of the lobes of the liver. Okay, I hope it is clear. Again, if it is not clear, liver is made up of lobes. Each lobe contains lobules. Lobules are hexagonal structures. These are polygonal structures. So we are making larger polygonal structure of lobule of the lobe of the liver. So from the each corner, a tubular empty space which is called as sinusoid enters towards the central, where the central vein is present. This is the central vein which I demarcated with the red pen. Okay, now these portal vein and hepatic artery which are entering into the liver reaches to each of the lobule, and it is entering the oxygenated blood through the hepatic artery and lesser oxygenated blood or deoxygenated blood through the portal vein inside the sinusoid. I hope it is clear. In between the two lobes, in between these two corners or two sinusoids, there is plate of hepatocytes. Plates of hepatocytes or hepatic cells are present. Plates of hepatocytes, correct? Or we can say hepatic cells are present. In between the sinusoid plates, means cells are placed one above the other to form a plate-like structure we called as plate of hepatocytes, or we call it as plate of hepatic cells are present. Now this is a sinusoid. Remember, sinusoid is a porous structure. Sinusoid is a porous structure, means from the sinusoids some of the material can move out towards this plate of hepatocytes, and some of the substances can move inside the sinusoid. This space between the hepatocyte plate and the sinusoid membrane is called space of Dizzy. Space of Dizzy. Now, when the blood moves, blood of hepatic artery, blood of portal vein moves, it actually passes into the space of Dizzy. The movement is going downward towards the central vein. So it is called a centripetal circulation. Centripetal circulation. So when it is going down, some of the material also come up and some do not move. That unmoved material or plasma moves through the membrane of sinusoid, which is porous in structure, towards the hepatocyte plate. The space between the hepatocyte plate and the sinusoid is called space of Dizzy. Through this space of Dizzy, the plasma enters into it. Remember that this is very, very important. On the other side, this is hepatocyte's one space, which is called space of Dizzy. On the other side, these cells also produces bile, which comes towards outside, which comes up towards the outside. This is the bile which is secreted by the lobule or hepatocyte cells. So bile comes out in opposite direction. This bile is taken out by another duct called bile duct. Also taken out by another duct or a channel or a portal called as a bile portal or bile channel. But this plasma which has entered towards the space of Dizzy also moves out, and this is lymph which is being created. This is a lymph which is being created. So lymph comes out through a channel of lymph. Previously, the biologist only observed the portal vein, hepatic artery, and the bile channel. 3 of these channels were observed, and they call them as triads. But afterward, biologists find another portal which is taking out the lymph. So this is a lymph channel. So previously it was called as a triad or portal channel. Together they all are called as portal channel. This is also called as a triad channel or triad portal. But afterwards, when we find the lymph is also coming from the channel, it is now called as a tetrad. Tetrad channel or tetrad portal. Together we call it as a portal. So once again, these lobule structure are the basic structural and functional unit of the liver. These are polygonal, and towards every end a sinusoid empties, tubular structure enters towards the centrally located what? Centrally located this central vein. These sinusoid membranes are porous. Through this channel, what is entering? Entrance is of, again remember, hepatic artery taking the oxygenated blood towards this sinusoid, and there is portal vein which is taking deoxygenated blood towards the sinusoid. So as it is entering inside, it is called as a centripetal circulation. But there is a space between the hepatocyte plate which is present between the two sinusoid and the membrane of the sinusoid. It is called as space of Dizzy. When the blood moves towards this central vein, some of the blood moves backward also and some gets stable. Through this, plasma enters into the space of Dizzy, right? And this exchange is taken between this exchange of plasma or material is through the blood is taken between the hepatocytes and hepatocyte plates and the sinusoid through this space of Dizzy. On the other side of hepatocyte, their bile secretion occurs. The bile secretion comes out or drained out. This is going inside, but the bile is coming outside. The same way, the plasma which converted into lymph also comes out from these lymph vessel. These two actually are coming out. This make centrifugal circulation. Centrifugal circulation. Correct? So remember, this is the circulation inside the lobes of liver. So we say, now this is, if we call as centrally located venous, so it is also having centrally located venous inside over here, inside over here, inside over here. Many lobule structure are there. These all the centrally located venous, central venous system, या central vein is of all the lobules get collected to form a lobule vein, and ultimately it forms a hepatic vein which enters into the inferior vena cava. So blood enters into the liver through hepatic artery and portal vein into the lobes of the liver. There it gets circulated by channels which previously were called as triads, now we call as tetrads. Now after supplying the blood from each centrally located central vein, they collectively form hepatic vein. It takes the blood after circulating into the liver to the inferior vena cava. Inferior vena cava, which opens into the right auricle of the heart. I hope it will help you to understand internal circulation in the liver. Thank you. If it is helpful, subscribe.